Good morning to everyone, and good afternoon, I guess, for those of you who are in Europe and maybe who knows where else. In fact, it would be kind of fun, I thought, if people wanted to put your country that you're coming to us from in the Zoom chat, especially if you're not in North America. It would be kind of fun to see where everybody's coming from. Um, Welcome to the 2020 Emsel Multiscale Microbial Dynamics Modeling Summer School. We are super excited to have everybody here. We have about 300 and some people online today, and I know we have almost 500 registered for the course. So it's super exciting for us to be here with you. And we're going to get started with just a welcome introduction, and then we'll get right on into the class. So we'll get a little, as we go through the introduction, you'll get a little idea of how this is going to be working. We'll give you some logistical information. So I'd like to start by introducing myself. My name is Tim Scheibe. I am a scientist at the Pacific Northwest National Laboratory in Richland, Washington in the United States. We're up in the Pacific Northwest region of the United States, so near the Pacific Ocean and near Canada. And I'm actually a hydrogeologist by background, and so I've worked for a long time in subsurface hydrology and computational hydrology, but with a lot of emphasis on biogeochemistry. So I've had a lot of exposure to microbes and people who work with microbes, which has been a lot of fun for me. And I'm going to let Nancy introduce herself in a second as well. Actually, why don't you just go ahead and quickly introduce yourself, Nancy, if you can.
Sure. Yeah, I'm Nancy Hess, and I also work at the Environmental Molecular Sciences Laboratory. I'm a geochemist by training, and I'm just so excited to meet all of you and have you participate in this summer school. So go ahead, Tim.
Excellent. So in some ways, I think myself and Nancy kind of represent what this class is kind of about, in the sense that I think Nancy is, more of a classical geochemist and a molecular scientist. Both of us work in the Environmental Molecular Sciences Laboratory, or Emsel, which is a user facility hosted at Pacific Northwest National Laboratory. I come more from the larger scale side of things, from the ecosystem scale, I guess you could say, or the aquifer scale. And really what this class is about is merging those two perspectives together, is learning how to take molecular scale knowledge and information, particularly microbial and environmental chemistry information, and incorporating that into predictive models of ecosystem function. So Nancy and I are your hosts for the meeting, but there's a huge range of people that are involved in putting this class together. And a lot of people have put a huge amount of work. I really want to call out a lot of the organizations. You'll hear a lot about KBase today. That's the DOE Systems Biology Knowledge Base, and a large team of folks have put together a really great set of narratives in KBase for us to walk through. The Joint Genome Institute is a partner user facility to Emsel that's located in Berkeley, California, but obviously by their name, the JGI does a lot of work on genomics. The ESS Dive is a partner of the community in providing data repository. And we have groups from a variety of universities that I'll speak to that have put a huge amount of effort into bringing this to you. So really appreciate all of the work of the team that's put this together, and want to give a lot of credit to them. As we go through today, let's see if I can get this thing to go. Okay, so the course is co-sponsored by the Environmental Molecular Sciences Lab, which Nancy will tell you a little bit more about in a second, and also by the Subsurface Biogeochemical Research project at PNNL. And our project is focused on river hydrology and biogeochemistry, and I'd invite you to learn a little bit more about the project through the URL that's listed here on the left. And one important aspect of this project is the Wonders Consortium that you'll be hearing about in the next presentation from James and Amy. And I'll let Nancy describe a little bit about Emsel.
Sure. So Emsel is an Office of Science user facility. It's funded by the Department of Energy and managed by the Biological Environmental Research Program. And as Tim mentioned, we're located in eastern Washington State, kind of that larger red dot in the map over on the right-hand side. And it's really a wonderful user facility, and we have extensive expertise and capabilities in both the environmental and biological science areas, and we work with users from all over the US and actually the world. And we collaborate them on research projects that in an integrative environment where we combine the experimental modeling and computational approaches to really span that molecular to ecosystem scale science that Tim was talking about earlier. And we're able to work with users based on a peer review proposal process. And so you can check out the link there underneath the map and learn more about that. But the really exciting part is once your proposal is accepted, we're able to collaborate with you without cost to you. So an additional supplement to your research, and really expanding the research that you can do probably beyond the resources that are available in your own institution. And it's a great learning environment, so you don't even have to be an expert when you come to Emsel to try something different to advance your science. And again, just to echo what Tim said, this particular workshop is really trying to understand those metabolic pathways that link microbial activities to ecosystem function. And this is enabled by capabilities at Emsel that you'll hear about, but also JGI, the K-BASE program that enables a lot of this modeling, and many other programs that are sponsored by the Subsurface Biological Research Program, SBR. So it's a wonderful opportunity to really see this integration of scientists from across the US using capabilities that have been provided to us by BER. So I'd like to thank the program managers for their support as well. And with that, Tim?
Okay, thanks, Nancy. So I'd like to just talk a little bit about the summer school and its motivation and logistics before we get into the first science presentation. So why did we decide to do the summer school? As you think about ecosystem science, a lot of the words showed in this word cloud are important to us. And as we've been going through the last, say, 5 to 10 years, a lot of other important words shown here have really become prevalent in science. Things like big data, exascale computing, open science, as well as computational biology, metabolomics, genomics, machine learning. And all of these things, we thought about how can we really put these together to address the problems that we have in ecosystem sciences. And a couple of years ago, back in the 2018 Fall AGU meeting, we held a workshop called Modeling Microbial Dynamics. It was involving many of the people that are involved in this summer school this week. And you can see some of those folks in this photo here that were panelists there. That kind of primed the pump for some of the thinking around these ideas, got people thinking that we could actually really come together as a community and go across these scales. We had a follow-up session at AGU last year, an oral and poster session on linking genes to terrestrial carbon and nutrient cycling. And that led to the idea to have this class as the second annual summer school at Emsel. We had a course last year that was on aerosol sciences. And this year we wanted to have one on microbial modeling. And our original plan was to have an on-site class at PNNL limited to about 25 students. And we had an application process in place, and we're doing that. But then, of course, the COVID-19 pandemic came around and forced us to go virtual with this meeting. So the benefit of that, however, is to allow us to open the course to a much larger audience. And I said here, more than 300 people registered. I made this slide a couple days ago. There's actually close to 500 people registered for the webinar, which is really super exciting. This is today's agenda, and I would like to point out that there are on the agenda there are a number of places where there are things listed like extended breaks or early adjournments. And what we're doing during this period of time is we did actually select 28 students from the applicants, and those 28 students are going to be doing some detailed hands-on exercises with the instructors in private sessions during those times. So we still have that opportunity for those students to get a close hands-on experience and go a little bit deeper with the data sets that we'll be sharing. But we have the chance to share most of the presentations and tutorials with the open public here, which is really great. And we're very excited that you're here. So when you see those kind of things in the seminar, you can know that's what's happening. I think we have a super exciting agenda and an amazing faculty presenting to you today and throughout the week. Today, we're going to focus on the topic of metagenomics. And that's really how genomes are measured for communities of microorganisms in the environment. And then Tuesday, we'll talk about metabolomics, or the character of organic products, organic matter in the environment, and how that affects microbial functioning. And then Wednesday, we'll get into some metabolic modeling at the scale of individual organisms and communities. And then finally, Thursday, taking that into reactive transport modeling. And so that's kind of the pipeline we're going to walk through with the students with some specific data sets. On Friday, we hear from a number of speakers talking about science applications in different ecosystem type of settings, ranging from river systems, watersheds, to marine systems and wetlands and so forth. I did want to point out here a number of our university collaborators. We have a group from Colorado State led by Kelly Wright, and that's been instrumental in putting together many of the sessions here, as well as Hyun Sab Song's group from University of Nebraska-Lincoln, leading a number of the tutorials as well. And then we'll have presentations from professors from the University of Georgia and University of Massachusetts as well. So we're excited to have all of those folks with us. This slide gives you a really dense overview of what the summer school looks like. And so I don't expect you to memorize this, but the basic idea is we're going to have some data sets that were collected by a community effort called Wonders, and you'll learn about that very shortly. And those community data sets were collected in seven different settings. And so we have 7 student teams working on those throughout the week. We'll be walking through those data sets, which include metagenomic and metabolomic information, and taking those through a variety of different tools, many of those embodied in the KBase system, which is, again, the DOE Systems Biology Knowledge Base. And others, a few of those not directly in KBase, but most of them in the KBase narrative system. So you'll really learn how to use KBase, how to walk through these different processes, all the way from raw data at the molecular scale up to a reactive transport model. And the example we'll be using is a simple 1D column type of model. And then again, in the last day, we'll hear about a number of large-scale applications of those kinds of models. I would like to point out up here in the right is the link to the KBase website. If you have not done so already, I would strongly encourage you to go to the KBase website and create a user account there, and you'll be able to then follow along with many of the narratives that you'll be hearing about in the tutorials later today and throughout the week. So the course objectives really for the entire group is to learn about recent advances and new tools that exist or have been developed recently to integrate what we would call omics data. So genomics, metabolomics, transcriptomics, all these different kinds of molecular scale biological data into environmental reactive transport models. To also learn how to access a number of resources that are provided to the science community by the U.S. Department of Energy, including our user facilities like Emsel and JGI, data resources like ESS Dive, and software tools such as KBase, pFlowTran, and other things. And then interact with other interested scientists in a multidisciplinary forum to develop relationships and networks. And part of this is for the students and the participants, but part of this is also for the team of instructors that as we've worked together to prepare this for you, I think we've also developed stronger collaborative relationships, and hopefully those will carry through into better science in our community. And then for the student teams that'll be working in the private sessions, they'll be performing hands-on analysis of these actual environmental data sets from Wonders under the supervision of the instructors. And they'll be addressing some specific science questions with their teams and between and across the teams. And hopefully our goal is to develop some preliminary scientific results that can be used for AGU abstracts and even some journal manuscripts. So just to catch a few logistics, the public Zoom meetings, such as the one we're in right now, are being run in webinar mode. So that means that only people defined as panelists or instructors are able to turn on their video and audio and respond to chat. As the rest of the participants can submit questions through the chat, those will only be seen by the panelists. And we have some moderators working with us that are monitoring those chat windows and will transmit those questions to the instructors during the set question and answer times. But since that webinar mode is kind of restrictive for the participants, in order to facilitate more interaction, we've created what's called a Discord server space. And if you're not familiar with Discord, it's an environment somewhat similar to Slack, which you might be familiar with. The link is here, and I would encourage you, if you haven't done so already, go ahead and type that link into your browser and join Discord. And I'd also say, if you can, change your nickname to be your full name using the pull-down menu at the top. You don't have to change your username, but if you can change your nickname, then that shows everybody who you are. At that point, you can also include your preferred pronouns if you would like to do that, as several have. And the text channels in Discord are for chat, and they're for open chat for anybody to use. There are also voice channels that can be used for live interaction. So when you go into one of the voice channels, you can actually turn on your audio and video and have live chat with people. And there's a whole bunch of different areas. During the public presentations, I would suggest that you use the plenary session channel and the chat channel to chat and to ask questions. And again, this channel will be monitored during the sessions by our moderators, and those questions will be passed back along to the speakers. Again, wanted to remind people to register for an account at KBase to follow along with the narratives that we'll be going through during the tutorials. So special thanks to our support team. We have some communication staff here from P&L that are observing, and they'll be developing some material for articles and social media and posting throughout the week. And so you can talk to Linda, Andrea, or Corey over in Discord if you want to meet them, and if you have any ideas for articles or social media postings. If you are posting on social media, we encourage you to use this hashtag. And our moderators are Becky Ford, Linda Isaacson, and also Sonia Enlow is helping out with that. Sonia's been the project coordinator helping organize this whole thing. And Sarah Heilman is working as our IT coordinator running Zoom for us. If at any point you have a problem with your connection or you don't know what to do and you have you're kind of at wit's end, Becky has given her cell phone number. For those of you outside the US, that would be +1 as the country code, and then area code 509-528-6741. And Becky's available to help you troubleshoot if all else fails. So thank you very much for your attention and for your participation today. We're super excited to meet you all and to hear about your exciting research and all the things that you've been doing, and to share some of these tools and ideas with you throughout the week. Look forward to talking with you over in Discord. And with that, I'm going to turn it over to the next speaker.